In the fungi, we have studied about the general characteristics, the thallus organization and the reproduction methods in fungi. And related to that, we have studied about the life history of Rhizopus and Mycelium. So, after studying the life history of the Mycelium and Rhizopus, we shall now study about the life history of Puccinia graminis. So, Puccinia graminis is a fungus which is a obligatory parasite. Now, this Puccinia graminis belongs to the group Basidiomycetes, and it is a obligatory parasites which requires the living host plant. So, on a host plant, the Puccinia will live and reproduce. And however, when we say that it is a obligatory parasite, so parasite means definitely they are usually pathogenic. Similarly, or the same way, Puccinia graminis, which is an obligatory parasite, causes a kind of disease on wheat called as the black rust, or sometimes it is called as the stem rust, because it affects the stem and leaves, and the disease is called as the black rust or stem rust. There are different species of Puccinia which cause different types of rust disease. For example, Puccinia striformis, it causes a disease called as yellow rust, or sometimes it is called as the stripe rust. That is, the disease is in the form of stripes. And there is another Puccinia species like Puccinia recondita, which cause orange rust or leaf rust. So, like that, orange rust or leaf rust disease is caused by Puccinia recondita. Now, usually the symptoms of this rust, whatever may be the type of rust, whether it is stem rust, that is black rust, or orange rust, or this stripe rust, or the leaf rust, then the yellow rust, or the leaf rust, whatever it is. Now, this rust disease, there is the appearance of what are called as the pustules. Pustules are some inflamed regions on the surface of the stem and leaves. So, these pustules, they are just like pimples, what we see. So, pimples means there are some swelling on the surface of the skin. Same way, pustules are such structures which are seen on the leaves and stem, and they are somewhat rusty in appearance, and they are of different colors like black or sometimes yellow color or like that. That is the symptoms of this disease which is seen. In case of stripe rust, what happens? These pustules are in the form of strips. Though they are swellings, they are in the form of strips, but in the other cases they are of somewhat round or the different spherical patches which are seen on the leaves and the stem. Now, coming to the structure of the mycelium and how the reproduction takes place. So, here the mycelium, it is a septate. We have studied in the fungi that Phycomycetes is the group which is aseptate fungi, example Rhizopus, where the mycelium or the hyphae is cenocetic. But in the Ascomycetes, Basidiomycetes and Deuteromycetes, the hyphae is septate. So though it is a septate, the septa, the cross walls, the walls in between, they are having a central pore so that the nuclei and cytoplasm pass through it and enter into the neighboring cell. So here the hyphae is a septate, and the septate hyphae which is found in this Puccinia graminis is again of two types. Now as it is parasite, so it is found in the wheat, leaf of the wheat or the stem when it is young. So the mycelia or the hyphae, they are intercellular. They grow in between the cells. They are branched and they are septate and they are well developed. They are endophytic, what we call it as. Endophytic means growing within the organ, and they are localized in that particular organ like leaf or stem. So this hyphae is a septate. So as usual it is having a central pore there, and here there are two types of mycelia or hyphae, monokaryotic and dikaryotic. So certain hyphae, they are uninucleate, monokaryotic. There are certain hyphae bikaryotic. So in the same body we don't find both. So it is either dikaryotic or monokaryotic. So it is having actually both type in its life cycle. Now this dikaryotic mycelium is present whenever the Puccinia graminis, or when this mycelium is present in the wheat. So in the case of wheat host plant, when the Puccinia graminis is present in wheat plant, there the mycelium will be dikaryotic, and this mycelium is called as secondary mycelium. So dikaryotic mycelium is also called as a secondary mycelium in Puccinia graminis when it is in wheat. But however, this Puccinia does not complete its life cycle in wheat host only. It also requires another host called as berberis plant. So in barberry plant, it will complete its life cycle. So when the Puccinia graminis, or when this fungus is present in the wheat, it causes the disease like black rust and black rust or the stem rust. So when it reproduces there, next it will enter into the next host, that is barberry plant, barberis. So when it enters into the berberis plant, so there the mycelium becomes monokaryotic. The dikaryotic mycelium which was there in the wheat plant, so when it enters into the berberis plant, it will become monokaryotic, and that is called as the primary mycelium. So here there it will complete its life cycle. So that is why Puccinia requires two hosts to complete its life cycle. So when it is in the wheat plant, it is dikaryotic. That means it is called as secondary mycelium. But whenever it is in the berberis plant, so there it is monokaryotic. So it is called as a primary mycelium. So the wheat is a dikaryotic, the berberis is monokaryotic, and thus it requires two hosts to complete its life cycle. So such a type of fungi is referred as heteroecious fungi. So it is called as heteroecious fungi, and this phenomenon is called as heteroschism. So this phenomenon is referred as heteroschism, and this fungi is referred as heteroecious fungi. So because it requires two hosts to complete its life body. When it is in wheat, it is secondary mycelium, dikaryotic. When it is in berberis, it is monokaryotic and it is called as the primary mycelium. So this is what is heteroecious fungi, or what is heteroschism. That is that phenomenon what we see in the Puccinia graminis. Now when we shall see how the reproduction takes place in the host, that means in the wheat and as well as in the this berberis plant. Now when it is in the wheat or berberis, in both the types, in both hosts, Puccinia will produce different kinds of spores like Uread of Spores, Telospores, or what are called as Tubulator Spores, they are also referred as. Then there are spores which are called as Picnium Spores or Picnidio Spores, or sometimes they are called as Spermatia and Asio Spores or Asio Spores. Then Teleospores or Tubulator Spores and Basidio Spores. So these different kinds of spores are produced by Puccinia in both the hosts, in wheat and the berberis plant. Now we shall see what type of spores are produced in the wheat and what are produced in the berberis plant, and in which structures they are produced. So when the mycelium is present in the wheat, it is a dikaryotic. And this dikaryotic mycelium or secondary mycelium, it will produce the Uraid of Spores first. Now these Uraid of Spores which are produced, and again here the fungus which is producing different kinds of spores or many kinds of spores, it is called as polymorphic fungus, or it is called as polymorphism. So Puccinia is polymorphic fungus. Why? Because it produces different kinds of spores like Ureter Spores, Tubulator Spores, Picnium Spores, Picnidio Spores, then Spermatia, Asio Spores and Basidio Spores. Hence it is called as polymorphic fungus or polymorphism. And Puccinia is also called as heteroecious fungi because it is having two types of mycelium, and that two types of mycelium is seen in two types of hosts, and hence it is called as the heteroecious fungi. So as it completes two life cycles, or it completes its life cycle on two hosts with the primary and secondary hyphae, hence it is referred as heteroecious fungi. And it is also called as polymorphic fungi, polymorphic fungus, because it produces various types of spores in its life cycle. Now this hyphae, whenever it is present in between the cells, when it is intercellular, it will absorb the food material from the cells by the help of the hyphae which is branched in between the cells. Now the first structures which are produced in the wheat are the Uread of Spores. Then from these Ureter Spores, the Tubulator Spores are produced. Now what are these Ureter Spores? So the mycelium of the Puccinia is a dikaryotic in the wheat plant. So when it is a dikaryotic, so these hyphae are in between the cells, intercellular, that is between the two epidermal layers, that means within the host epidermis. The hyphae are within the epidermis, in between the cells, intercellular. Now when it is there. Now these, suppose this is the epidermis. The hyphae are present inside. Now this hyphae, when it starts reproduction to produce Ureter Spores, some of the hyphae, they grow out vertically above the epidermis. So when they grow vertically above the epidermis, these vertical hyphae which grow above, suppose this is the hyphae which is growing, these are the branches of the hyphae. So these are called as the Uradosaurus sporophores, they are called as. They are called as sporophores. So sporophores are the vertical hyphae which originate above the epidermis, and when they grow vertically, so they are called as the sporophores. Now the terminal cells of this, suppose these are the terminal cells. So these terminal cells of the sporophores, they are called as the Uread of Spores. So the Uread of Spores, they develop. So these are the Uread of Spores which are developed there. So the terminal cell, so they are septate hyphae. So the terminal cell of the sporophore above the epidermis, so they, it is called as the Uradospore. So these Uradospores which are liberated, so this structure is called as Uradosauri. So the group of the Uradospores, they together are called as Uradosaurus. This is Uradosaurus or Uradosauri plurally, and that appears like a what is called as a pustule, a rusty appearance. And these Uradospores which are found, they are liberated by the separation of the epidermis or above the epidermis. So they get separated. Now these Uradospores are somewhat oblong. Oblong means somewhat elongated, not exactly. So that is, they are broader, but they are somewhat elongated. So these Uradospores, they are dikaryotic, because the mycelium is dikaryotic. This is the cell. These are the septa which are seen. So the terminal cell of the vertical hyphae, the terminal cell of the sporophores, they develop into the Uradospores. And these Uradospores, they separate. And these Uradospores, they are somewhat oblong, means broader and elongated, and of course they are dikaryotic. Now the Uradospores are having two walls. So the outer wall is the exosporium and the inner wall is the endosporium. So there is one outer wall. Suppose this is the outer wall. So this is the exosporium. So this exosporium is somewhat spiny in nature, means it is having a pointed structure, whereas endosporium is smooth. So endosporium is smooth, and whereas the exosporium is somewhat thick and it is having spiny like structure, which is called as acinulate or acinate. So this is the structure of the Uradospore. And these Uradospores, they are having small pores on its surface in the central region. So there are 4 germ pores. So these are the germ pores there. So there are 4 germ pores in the equatorial region of the Uradospore, from which the germination takes place. So the germ pores are arranged in the equatorial region, and then the Uradospore germinates when it falls on suitable conditions. So this suitable conditions are nothing but the leaf only. So they may produce on the same leaf or another leaf, wheat. So when they fall on a suitable medium, the Uradospores will germinate and produce further hyphae. And this hyphae which is growing. Now suppose this is a Uradospore. Just imagine. Now this Uradospore will produce the hyphae like this and branched hyphae in this way. Now this enters into the stomata of a leaf. So the hyphae will enter into the stomata and it will form a bulged structure which is called as appressoria. So it will penetrate into the stomata, or it will grow into the stomata and bulge to form a structure called as appressoria, and this appressoria will develop an enlarged structure, a peg-like structure, and then it will enter into the leaf, and then it will further branch in between the cells. That is what the Ureter Spores will further develop. Now after the development of this hyphae from the Ureter Spore, so this hyphae or the mycelium produces what are called as the Tubulator Spores. The Tubulator Spores are developed in the wheat leaf. So these Tubulator Spores, they are also produced in groups in the same way. And the Tubulator Spores are produced in the epidermis. So as the hyphae is present inside the leaf in between the cells, intercellular, so it will also grow and produce what is called as the teleal stage. So this stage is called as the uradial stage when the Uradospores are produced. So when the Uradospores are produced, it is the uradial stage. But when the Tubulator Spores are produced, it is called the teleal stage. Now in the teleal stage, where the Tubulator Spores are produced, so these Tubulator Spores are again produced on the surface of the leaf, and they are also exposed on the surface. So like this, these are the Tubulator Spores which are produced on the leaf like this. So these are the spores which are produced. They are also septate. And the Tubulator Spores, these are somewhat irregular in shape, not exact shape they are, and they are somewhat elongated, and they are dark brown or sometimes black in color. And this Tubulator Spores, they are developed in a structure called as Tubulatosauri, Uradosauri, Tubulatosaurus. So the Tubulatosaurus will release the Tubulator Spores, and each spore it is having two cells. Now suppose this is a Tubulator Spore. It is having two cells like this. So it is a two-celled structure. This is a binucleate, dikaryotic, single cell dikaryotic, Uradospores. But Tubulator Spore, it is made up of two cells, and again it is somewhat dark brown in color. So that is the Tubulatosauri. Now these Tubulatosauri or the Tubulator Spores, they are having two cells. So there is one germ pore at the tip of this, there is one germ pore by the side of this. Here there are four germ pores in the lateral region all around cell. But here there are two cells. One cell is having at the tip of here and another cell on the lateral side. So the germ pore is the region where the cell will grow out into the mycelium or the hyphae. And then the germ pore, which is one at the apex in the upper cell, and in the lower cell it is present on the lateral side. So this the Tubulator spores, when they fall on a suitable medium, so under favorable conditions, the germ pore, each cell will develop a structure which is called as the promycelia. So this forms a structure called as the promycelia, like this. So this is called as promycelia, or sometimes it is also called as another name for that, the basidia. It is also called as basidia. Basidia they are called as. So the basidia are produced from each cell through the germ pore. Now each basidium, so basidia is plural, so each basidium contains a diploid nucleus. So that means the diploid nucleus in the basidium, it divides to form, means by mitosis it will divide and gives rise to this 4 cells. Means actually the nucleus here, it divides by meiosis, not mitosis, to produce 4 nuclei. So this divides by meiosis, this also divides, and these nuclei will move into the basidium and they form 4 basidiospores like this. Suppose this is one basidiospore, this is one another basidiospore, this is another basidiospore, this is another basidiospore. So like that the basidiospores, they are formed on the basidium, and we find these haploid basidiospores. So these are basidiospores which are produced, 4 basidiospores. Now out of these 4 basidiospores, 2 are of plus type and 2 are of minus type out of the 4 basidiospores. So the basidiospores are haploid, and each basidium produces these 4 basidiospores which are born on the structures which are called as stalk-like structures called as sterigmata. So on each sterigmata, the basidiospores are produced. So basidiospores are produced from the development of the Tubulator spores. So this is the next stage in the wheat plant. Now the basidiospores are developed on the sterigmata which are uninucleate, and each basidiospores which developed, they are released from the sterigmata, and out of the 4 basidiospores, 2 are of plus type and 2 are of minus type, and the basidiospores, they are separated from the sterigmata, and when they fall on a suitable medium, then they germinate into the next stage. So they are carried to the next host. So that means till this, the development is seen in the wheat plant, the Uread of spores, the Tubulator spores and basidiospores, all these 3. But the basidiospores will not develop on the wheat plant, but they will only when they fall on the next plant, that is barbarous plant, that is they will develop into the next stage, that is the Picnium or the Picniospores. So in the barbarous plant, or berberry plant, there the Basidiospores develop. In any case, the Basidiospores will not develop on the wheat plant. Now, when they fall on the epidermis of the leaf in the berberis, they develop into the Picniospores. Now, these Picniospores, they are the structures which are produced from the Basidiospores. So, after about 4 days later, when the Basidiospore fall on the epidermis, so this Basidiospores germinate and produce monokaryotic mycelium. That means already we have mentioned that the dikaryotic mycelium or the secondary mycelium is produced only in the wheat plant, whereas the primary mycelium or the monokaryotic mycelium is produced in the berberry plant or the berberis plant. Now, each Basidiospore, when it deposits on the wheat or sorry on the this leaf of the berberis plant, it will give rise to the mycelium and produce a structure that is a monokaryotic mycelium. It produces a structure called as this Picnium. So, this Picnium, suppose this is the epidermis. So, the Picnium is produced inside, and there are a layer of cells at the base of the Picnium like this. So, this is just an diagrammatic view what I am showing here. So, this is the structure of the Picnium where the Picniospores are produced. So, Picnium is a this flask shaped structure. So, this Picnium develops into a flask shaped structure, and at the base of the Picnium there is what is called as the hymenium layer or the hymeneal hyphae. So, the cells of the hymeneal layer of this vertical hyphae, they are developed at the base of this Picnium. So, these vertical hyphae that develops of this from the hymeneum layer, so that vertical hyphae like this, suppose this is one vertical hyphae, this is another vertical hyphae, like this the structures are produced which are called as Picniosporophore. So, this is called as Picniosporophore, that is the structure. So, this Picniosporophore, it gives rise to the Picniospores at the tips in the form of basigenous chains like this. So, these are called as the Picniospores, which are produced in the Picnium structure that is flask shaped. So, the Picniospores, they are released through the opening at the end of this, which is called as ostiole. So, the ostiole is the mouth or the opening of the Picnium through which the Picniospores are released. So, these Picniospores, they are produced in chains which are called as the basigenous chains. And when these Picniospores, they are, I said, they are monokaryotic already, but dikaryotic cells are seen only in the wheat plant. So, the monokaryotic cells, they are produced in berberis plant or barberry plant, and then the Picniospores, they develop into the Asio Spores. Now, these Picniospores or the Picnium, they are having certain structures at this region. There are certain hyphae here which are called as the flexious hyphae. So, they are called as the flexious hyphae, and there are certain extra protectional hyphae which are called as the paraphysis. The paraphysis are seen on the lateral side of the Picnium, and then the paraphysis are just the extra hyphae which are found in the neck region of the Picnium, and certain vertical hyphae, they are fertile. So these fertile hyphae are called as the flexus hyphae, and they come out through the ostiole. And when the spores are released, these spores, they fuse with the flexious hyphae, and they become dikaryotic hyphae or dikaryotic mycelium. So as the spores are monokaryotic, uninucleate, the flexus hyphae is also uninucleate. So the spores, the pycniospores, they fuse with the flexus hyphae and form dikaryotic mycelium. So that means the flexus hyphae converts the monokaryotic mycelium into dikaryotic mycelium, on which the Asio spores are produced. So next we come across the Asio spores. So the Asio spores, they are also produced in the berberis leaf only. Now what are these Asio spores? So this Asio spores are produced in a structure called as acidium. So they are produced only after dicaryotization. Only when the flexus hyphae becomes a dikaryotic, then we call it as, then we come across the development of the Asio spores. So the Asio spores or the acidio spores, they are also referred as. So they develop from the hyphae of the acidial primordia. So the acidial primordia are one type of hyphae which gives rise to the Asio spores, and they are also called as protesia. So this protesia is one type of hyphae which is developed, and that gives rise to the Asio spores. So the Asio spores, they are produced again in the leaf itself only. That is, it consists of the dikaryotic mycelium. So this acidial primordia or protesia, which is produced from these flexus hyphae, it is also surrounded by monokaryotic hyphae. So that monokaryotic hyphae is somewhat having a pseudoparenchymatous structure, and these dikaryotic cells of the acidial primordia, they develop into vertically elongated cells on the lower part. So vertically elongated cells are produced like this. So certain cells also are produced on the either side. So these vertically elongated cells which grow downwards in the stromatic layer called as hymenium. So it consists of Asiospore mother cells. So these are Asiospore mother cells. So these Asiospore mother cells, they give rise to the Asiospores. So how they give rise to the Asiospores, we shall see. So the Asiospores which are developed from these Asiospore mother cells. Now some of the cells which are produced at the periphery, they do not develop into Asiospore mother cells, but they act as the protective layer, and that layer we call it as the peridium. So the peridium is the cells which are produced from the periphery cells, and within this the Asio mother cells are present, and they divide and gives rise to the Asio spores. How they divide we shall see. So the Asio mother cells, they elongate and they divide into two cells. So when they divide into two cells, suppose this is the Asio mother cell. So when it divides, it forms a large cell and a small cell like this. So the division is unequal, in other words. So the larger cell which is divided, it becomes the Asio whereas the smaller cell which is formed, so this forms the Asio and this small cell forms the intercalary, or it is also called as the disjunctor cell. So intercalary cell, or it is also called as a disjunctor cell, it is non-functional as it is a small in size. So this is a sterile in nature, means it does not involve in the reproduction further, but the Asio spores, they involve in the further development. And then when these Asio spores, these intercalary cells, so from each Asio mother cell there is a division such that a large cell is produced, a small cell is produced. The large cell will become the Asio the small cell will become the intercalary cell or disjunctor cell. So this Asio separates by the degeneration of the disjunctor or the intercalary cell. So when the intercalary cell degenerate, the Asio become free, they separate, and the peridium actually, which is completely covering the Asio mother cells, it will rupture and release the Asio So when the Asio are released, they are carried, and the ascidium, this structure which is called as ascidium, so it appears like a ball, and this ball-like structure, when it is ruptured by the peridium, the Asio are carried by wind or sometimes by insects or even by water. So when the Asio are released, now each Asio is actually dikaryotic, remember, because the monokaryotic has become dikaryotic. So the division takes place. Suppose here there are two nuclei, each nucleus will divide to form two nuclei, so that means 4 nuclei are produced actually. So when the 4 nuclei means each cell is binucleate, dikaryotic. So the Asio is somewhat round in shape, and this Asio which is unicellular, but it is a dikaryotic, and also it is having a outer and inner wall just like this Uraid of Spore and the teleospore. So the outer wall is somewhat spiny, or sometimes it may be smooth also, and the inner wall, that is the endospore, it is smooth, and the spores are somewhat orange colored. So these are orange colored, and these Asio they are carried by wind or water, and only they further develop on the wheat plant. So that means till here it is the barberry as the host followed. So like that the Puccinia graminis fungi, which causes the black rust or the stem rust, it will require 2 hosts like wheat and berberis plant. Hence it is called as heteroecious fungi, and it is producing different type of spores, which we have already studied in the wheat plant. There are Ureter of Spores, and there are Tubulator Spores, and there are B.Sc spores. But in the barberry plant there are Picnium spores and there are Asio spores. So thus the Puccinia graminis is called as the black rust or the stem rust disease which is produced in the wheat plant. So this is about the life history of Puccinia graminis.